Hi, Michelle Glass here. Welcome to another one of our episodes for Chapter 7, Lecture 7. We are still talking about photosynthesis, and this video is all about that first major reaction that happens. It is called the light-dependent reaction. In the light-dependent reaction, we are going to see that our water molecule is going to be split so that we get oxygen gas given off, and we get a hydrogen that we can reduce NADP plus with. So that was kind of roundabout saying that we're going to pull hydrogen off the water molecule and add that to NADP plus to make NADPH. We are going to then use that as an energy molecule to help us in our Calvin cycle. We also see that this is the step where we are converting our light energy into ATP energy, and we will be using that also in the Calvin cycle. So we can refer to this as sort of our energy factory. We are making the NADPH and the ATP that we need for our second step in our photosynthesis procedure. All of this process is happening within the membranes of those thylakoids. Remember, we see stacks of thylakoids inside the chloroplast. We call those stacks grana for the plural form or granum for singular. Okay, now here's something we need to know about electrons. Electrons can be excited when they absorb photons or light energy. This can excite the electron. An excited electron has a lot of potential energy. What we will see is that excited electrons will real quickly drop. When they drop, they release all that energy that they absorbed as part of that getting excited stage. And if you allow that electron to just drop, you're going to get this big release of heat and light energy. So this is way too much drama to happen inside of the cell. So what we're going to see happening with our chloroplast, with the light reaction, we're going to see a very controlled excitement of our electrons so that we can harness all of that energy to use it to make our ATP instead of having it just released as light and heat back into the environment. We can have the energy harvested or harnessed using what's called a photosystem. So embedded in the membrane of the thylakoid, we will see there are two different types of photosystems. The photosystems are going to have several different pigments. Pigments are molecules that can absorb wavelengths of light, so they're absorbing photons. Those photons then are going to be exciting electrons, and we will see that we will be exciting electrons of our special chlorophyll A molecule. And we're going to do this in such a way that we can grab hold of that energy and have slow releases of that energy so that we can use it to make our ATP molecule. Okay, so here's our basic information. The photon is exciting electrons. Those excited electrons are being passed ultimately to our chlorophyll A, and then we're going to see an electron acceptor, which is going to accept those excited electrons, and then we will see it being passed through an electron transport chain, similar to what we saw in our cellular respiration discussions. And in that way, then we are grabbing hold of these small releases of energy and using it for work in the cell. I mentioned that there are two different types of photosystems. These have been named based on their discovery. So unfortunately, the naming is sort of backwards. We have photosystem 2 working first. That's where we're going to see our first set of excited electrons. Those excited electrons are going to get passed through an electron transport chain to our photosystem 1. And then we're going to see, at the end of these steps, our NADPH formed and also our ATP molecule formed. Okay, so just pay attention. It goes photosystem 2, electron transport chain, photosystem 1. All right, so remember we're looking at these processes occurring inside the chloroplast. We already talked about our chloroplast having the double membrane. It has those little stacks of thylakoids called granum. Each one of those thylakoid membranes contains our chlorophyll pigment. So we are seeing that it's the thylakoid and the chlorophyll that's giving the green color to the chloroplast and to the plant as well. Remember too, we have fluid surrounding the grana. That fluid is called stroma. Okay, so now we're zoomed in on our thylakoid. So you might want to try and draw a picture similar to mine. You might want to use the artwork that's already produced in your textbook. You might just want to kind of write things out in words, so that's up to you. So notice we have photosystem 2, then we have our electron transport chain, and then we have our photosystem 1. Also embedded in our thylakoid membrane, we have our old friend, ATP synthase. So remember, this is a protein that is going to phosphorylate an ADP molecule, making our ATP. And notice that the thylakoid membrane is surrounded by the stroma. Okay, so what we're going to see is in photosystem 2, that's where we start. So look at your photosystem 2. We're going to see that those pigments are going to absorb photons. This is going to excite electrons. Those excited electrons are going to be passed down to those special chlorophyll A molecules, and then the electrons are going to pop off those chlorophyll A to an electron acceptor. In order to replace the electrons that are excited, we're going to see that enzymes will split a water molecule into hydrogen and oxygen. So this is the step that gives us our oxygen gas, which leaves the chloroplast, leaves the cell, leaves the plant, and goes out into the atmosphere. That hydrogen is split so that we get a hydrogen ion, and we get the electrons, which are going to go up and replace those excited electrons from chlorophyll A. So we need to replace those electrons that have been excited. The electrons that were excited in our photosystem 2 are going to be passed through the electron transport chain. The energy that's released as those excited electrons get passed down will be enough energy to pump hydrogen from the stroma into the thylakoid space. And then in the space, you have a high concentration of hydrogen ion that then moves through that ATP synthase, causing it to spin, phosphorylating ADP, creating our ATP. So that's how we're going to get that ATP molecule produced. All of that hydrogen that's getting pumped goes back into the stroma so that we're maintaining a high concentration of hydrogen ion in the stroma. Those electrons that were excited and were passed through the electron transport chain are going to now move to the photosystem 1. Notice that in photosystem 1, you have photons being absorbed, so you're exciting electrons there. Those excited electrons pop up to an electron acceptor. So all of that is just like what we saw in photosystem 2. You are using those electrons from the electron transport chain to replace those excited electrons from the photosystem 1 chlorophyll molecule. The electron that is excited in photosystem 1 is then going to pair to an NADP plus to make our NADPH. So we have now made the energy molecules that we need for our Calvin cycle. So going through it again, start back at your photosystem 2, that's our starting point. Notice that you have photons exciting our pigments, so exciting electrons in those pigments, and they're being passed from one pigment to the next, making their way to our chlorophyll, where that excited electron is going to then join with that electron acceptor. In order to replace the electrons from chlorophyll A, water molecule is going to split. Oxygen gas is given off out into the atmosphere, and then the hydrogen is divided into an electron and a proton. This electron from water is going to replace the photosystem 2 chlorophyll electron that was excited by light. The electron that was excited by light is going to be passed to the electron transport chain. This then provides little releases of energy that can pump hydrogen from the stroma into the thylakoid. That hydrogen is moving then through the ATP synthase molecule in order to phosphorylate ADP. So this is where we get our ATP production, and we're going to stick that hydrogen back out into the stroma so that we're replacing that concentration gradient. We see the electron from the electron transport chain is going to replace the excited electron from photosystem 1. In photosystem 1, you are absorbing wavelengths of light. You're exciting electrons from those pigments. The excited electrons from photosystem 1 are going to be used to reduce NADP+, forming NADPH, which is our energy molecule that we need along with ATP to do our activities at the Calvin cycle. So this, again, is our light reaction. We're splitting our water molecule. We're passing electrons from photosystem 2 to an electron transport chain to photosystem 1. During these steps, we're making both NADPH and ATP, which is going to make its way over to the Calvin cycle. We're making our oxygen gas in this step, which is going to make its way back out into the environment, into our atmosphere. And this is all happening, oops, big typo. This is happening in the thylakoid. And it involves our chlorophyll pigment molecule. All right, so stay tuned for details on the Calvin cycle, and that's it for now.